Hi, I'm Deb Agarwal. I'm the Deputy Task Area Lead for Modeling and Simulation within the NAWI project. And I'm really pleased that you've all joined. We're going to talk about the work that's been going on in the Proteus High-Fidelity effort, which is really looking at bringing high-fidelity CFD modeling to the task of looking at desalinization and water treatment. And actually, I want to turn it over to Ramanan to actually begin the talk. And if you have questions during the talk, feel free to put it in the Q&A or the chat actually. And then we'll also be opening up the mics for discussion at the end of the talk. So take it away, Ramanan.
Thank you. I want to get started by acknowledging my collaborators from this high-fidelity modeling task. We are a team spread across Oak Ridge National Laboratory, National Renewable Energy Lab, and also Stanford University. Today, you are going to hear from several of us, myself, Ramanan Sankaran, Vimal Ramanuj, and also Hari from NREL. Mark Day is also joining us on this webinar. I also want to take this opportunity to thank our advisors. One of the first things we did in this modeling and simulation high fidelity task was to identify challenge problems that we can go after. And on that, we got very good advice from our advisory committee, especially Eric Hogue, Jeff McCutcheon, and Tim Bartholomew. With that, I wanted to give a quick introduction into one of the first tasks that we did that, as I mentioned, was to identify challenge problems for our high fidelity modeling and simulation. We came up with two important challenge problems. Both were targeted at getting new scientific insights. Those are the two problems on which we have actually been working on in the last several months and where we are going to report our approach. We are going to report some of our early results and also discuss our future plans. The first of those is high pressure reverse osmosis, HPRO, that also closely relates to the task 6 of NAWI. The second challenge problem that we identified and that Hari is going to speak about a little bit later is the characterization of flow and pressure drop in complex geometry spacers. So you are going to hear from us on these two challenge problems and the recent advances or the recent work that we have done there. Now also, the high fidelity task sits within the modeling and the larger modeling and simulation task. And one of the key platforms that's being developed here is what we call the Proteus HF. And at this stage of the project, the Proteus HF consists of these 4 codes that are being exercised for challenge problems. The first is Quilt. Quilt is a flow solver from initially developed at Oak Ridge. Second is SUMS from Stanford. Third is NMAC foam, developed through NAWI by NREL and Stanford. And the fourth is EXA-MPM, a material point method code from Oak Ridge National Laboratory. We are going to get results from the first 3 of them and also discuss our planned work using the EXA-MPM as part of this presentation. With that introduction, I want to hand it over to Vimal. Both of us are talking on site from our offices here at Oak Ridge. And Vimal is going to present on our first challenge problem, the high pressure reverse osmosis.
Thanks. Yeah. Thanks, Ramanan. So let me also acknowledge the team members and contributors to this task, and also the challenge problem advisor. For the HPRO challenge problem, the major challenge is to address the membrane compaction and deformation at high feed pressures. This can lead to the support layer. So this can cause the support layer compression and increase the transport resistance. It could increase the risk of defect formation due to uneven compaction and may also lead to pressure drop in the permeate channel. At this point, the gaps in our understanding are basically how to model the mechanical compaction of the support layer and against the backing. How does that affect, how does the macrovoid pore geometry affect the membrane properties, and how does the compaction affect the transport properties of the membrane? And addressing these challenges would enable us to extend the RO process capabilities to even higher pressures and higher concentrations. So as a part of this task, we developed the following numerical modeling workflow, and there are basically two key links here. The first is the representation or the reconstruction of the pore scale geometries that could either come from reconstructed experimental images, or it could be synthetic or canonical pore geometries. These geometries can be exported to several physics simulation codes, some of which Ramana introduced earlier. And we can be looking at the compaction to mechanics models or pore scale transport simulations to characterize these membranes. Now, this workflow will enable us to gain more fundamental insights into the performance of the membranes, as well as develop better informed models that can be used for channel scale CFD and analysis. In today's talk, I'll focus on synthetic pore geometries as well as the pore scale transport simulations. And towards the end, we can come back and talk about the other key links here, depending on the interest. So the synthetic geometries are basically statistical representation of the pore scale structures. The Proteus HF workflow synthesizes these geometries that can be easily interactively visualized and compared against experimental images. These geometries are constructed within the code Quilt, and they can be exported in several different formats. It leverages several open source capabilities as a part of this HF workflow. Now, if you look at the first branch, these geometries are pretty lightweight, so they can be very useful for interactive visualization and collaboration with team members, and also for sharing with experimental researchers, so that we can easily and quickly identify some interesting geometrical features of the pore scale, and maybe also iterate on those geometries to construct a larger database. Now, these geometries are of course also fed to the physics simulation codes, such as the NPM code for compaction and Quilt for transport simulation. And depending on their end use, these files can be larger in size and can leverage the high performance computing platforms. So let me quickly go through some of the example geometries, parameterized synthetic geometries that can be constructed. Starting from the top left, we are looking at a multi-layer isotropic structure. Basically, this is a structure that has uniform spherical pores randomly distributed within a layer, and then multiple layers being stacked on top of each other. The second image shows a single layer graded structure. Basically, here is where we introduce a spatial dependence of certain geometric features. In this case, we are looking at the spatial variation of the pore sizes. Similarly, just like before, we can have a multi-layer graded structure by stacking different layers together. And on the right-hand side, the last figure actually shows the case where we introduce some other geometric features like the aspect ratio of the pores, and we can also introduce orientation of these these voids. So, and again, all of these geometric features can be randomized to obtain a specific type of distribution that is desired. And on the bottom, I've shown a 3D structure that represents elongated ellipsoidal pores, more or less trying to represent what is experimentally observed in some of the published literature. And I'll come back and talk about this geometry specifically after a few slides. The right-hand image, the right-hand movie actually shows captures the orientation and aspect ratio of these macrovoids along two different orthogonal cross sections within the membrane. So moving on to the transport simulations, these are performed using the code Quilt, which, as Raman said, is being developed here at Oak Ridge. It's an interface resolved flow solver. In the recent past, we have looked at several different applications. A couple of them I have highlighted here. On the left, we are looking at a multi-phase flow application where we studied a molten media reactor that's typically used for converting natural gas into carbon and hydrogen. And that is a really promising route because it does not involve production of CO2. On the right-hand side is a porous media application where we studied flow through catalytic porous packings. In this case, we are interested in looking at the effects of polymer particles porosity and how it affects the overall flow and chemical conversion rates in these applications. At the heart of this code build is what is called an implicit interface representation capability. This is done using a level set method. Just a quick overview of the level set. It is a signed distance function within a domain, and it's used to capture implicit interfaces embedded on structured meshes. So naturally, following the definition of level set, we can easily understand that the zero level set contour will capture the interface. And it being a distance function, it is also used to capture several geometric features like the orientation and curvature of the embedded interfaces. In the code, we have capabilities to represent dynamic as well as static interfaces. Since this particular problem relates to static membrane structures, I'll skip the introduction about the dynamic interfaces, but I can come back and talk about it later if there is interest. For this particular application, we chose the level set method specifically for two reasons. The first is that it gives a capability to model the pore deformations resulting from membrane compaction, and that could be from analytical or geometric transformations, or we could impose a deformation field that would result from mechanics calculations, for example, using an MPM code. Moreover, if you look at one of the experimental images I showed before, the membrane structures usually have two-scale porosity. They consist of large-size voids, which are called macrovoids, and the remainder of the membrane domain is made up of homogeneously distributed small pores. So with the level set framework, we can address the two-scale porosity of the membrane by fully resolving the macrovoids using the level set function, and the rest of the domain is represented using an effective medium model to address the transport through smaller pores. Now, in this case, we are interested in looking at membrane characteristics quantitatively. In particular, we look at the tortuosity, hydraulic tortuosity, permeability, and structure factor of the membrane. To analyze these quantities, we seek a steady state solution for the diffusion and flow transport in periodic domains. The flow in this case is governed by an externally applied pressure gradient. Now, going back to the synthetic membrane structure I showed before, our aim for this preliminary study was to first identify an experimental work that we can leverage to verify our numerical simulations results, as well as use these results to identify certain key parameters in the geometry or in the physics that we can explore later. So in the following slides, I'm going to show the calculation of membrane characteristics like tortuosity, hydraulic tortuosity, permeability, and structure factor based on a particular synthetic geometry that we have initialized following some experimental studies. Here is the work that was published a few years back by Manikam and others. This was actually a multi-layer membrane support structure, and we specifically identified one of the layers to create our synthetic geometry. Here are some of the parameters that are listed on the screen. Mainly, the membrane size in the thickness direction is about 150 micrometers. In the other two periodic directions, the domain is 4 times as big. The macrovoid cross section shows an aspect ratio of 4:1 in the cross section, and these are like about 400 micrometers in the elongated directions. And they also have a preferred orientation of about 15 degrees about the elongated direction. The overall porosity of this domain is approximately 43%. The following diffusion and transport simulations were performed on NREL's Eagle HPC facility. So let me come to the diffusion transport first. Here we performed two specific numerical experiments. In the first case, we apply a far field Dirichlet boundary conditions above and below the membrane. And based on the ratio of concentration gradient at the surface of the membrane, we can compute the effective diffusivity of the membrane structure. In the second experiment, we actually solved the steady state diffusion equation on a triply periodic domain, just like following the equations I showed a couple of slides before. On this right-hand side image, what we are seeing are the flux vectors, which basically, if you look closely, they actually represent tortuous diffusion flow paths that are resulting due to the arrangement of these macrovoids. Now, to quantitatively represent this effect, we calculate diffusional tortuosity by two different methods. The tortuosity is actually defined as the ratio of path length to the displacement between two periodic phases. And mathematically, that can be approximated from the flux vectors, like as shown on the left equation. For the non-periodic case, since we can compute the effective diffusivity of the membrane, we use the scaling law, like what is shown here in the middle, to obtain the tortuosity in that case. And in most membrane literature, structure factor is also an important quantity used to characterize the membranes. The structure factor in this case is defined as the ratio of porosity to tortuosity of the membrane. Now, we compared the numerical simulation results with what was published in the experimental paper I just referred to before. We observed a pretty good agreement between the simulation results and the experimental work. Although there are a couple of key points to be noted here. The experiments were performed using destructive testing and 3D image analysis, and these were performed for composite membrane structures. So those were multi-layer support structures, and then those were imaged to obtain the characteristics. The geometric characteristics I showed, the geometric features parameters I showed a couple of slides before, they correspond to the bottom support layer, which is where these characteristics are computed. But since these results are pretty encouraging, we are definitely motivated to start looking into multi-layer membrane structures, composite membranes specifically, and that will provide us even more fundamental insights into the performance of these membranes. Next, we looked at the flow simulations, steady-state flow simulations, and it is the flow in this case was driven by an externally applied pressure gradient across the thickness direction. It is important to note the K out parameter in this case. K out is basically the permeability of the unresolved space, basically the region outside of these macrovoids. And as we would expect, the flow behavior within this support layer, it strongly depends on the value of this K out parameter. For example, on the top image, where the K out value, or basically the unresolved permeability, is much high, we see that the flow is more or less aligned with the applied pressure gradient. And qualitatively, the macrovoids have minimal effect on the flow behavior. At the bottom, if we reduce the K out value by almost 2 orders of magnitude, we start seeing the effects of what is called as flow channeling. This is basically where there are localized high permeability flow paths existent within the membrane due to the arrangement of these macrovoids. And the flow basically prefers to go through these macrovoids rather than the unresolved space. In this case, we also observed non-zero transport in the transverse direction. And based on the average flow velocities in each direction, as well as the velocity vectors, we can easily compute the permeability tortuosity correlation, like what is shown in the top image here, top plot. Even more interesting is also the observation on the two charts at the bottom. Those actually show the tortuosity and the effective membrane permeabilities as a function of the unresolved permeability parameter. Now, focusing on the leftmost data point, on the right image, we can see that there is significant transverse flow when the unresolved permeability was very low. And in this case, actually, since we already know the orientation of the macrovoids, it can be theoretically shown that the tortuosity for this case would correspond to about approximately 3.8, which is what we would expect on the leftmost image here. At the other extreme, if we start increasing the unresolved permeability value, we can see that the transverse flow diminishes, and then and also the tortuosity approaches unity. This was an interesting analysis since it enabled us to verify the numerical results as well as identify some key geometric parameters in the synthetic geometry, like the orientation and aspect ratio of the pores, macrovoids basically. And we can further explore the performance of these support layers by creating several iterations of the synthetic geometries. Motivated from these results, these preliminary results, our next steps include studying the HPRO compaction using synthetic geometries, mainly by, this can be done by two different ways. We can either impose an analytical or geometric transformation model that will alter the level set function and basically change the shape of the macrovoids. Or we could also import the displacement fields obtained through mechanics calculation using the material point method that was initially introduced in one of the codes. We can also model the flow through these static compacted media and look at its effects on the overall transport properties of the membrane structures. And since we have the capability to generate several synthetic geometries, this also enables us to look at to analyze several of these membrane structures and try to explore the several pore structure characteristics of these membranes. So with that, I'd like to transition to Hari, who will talk about the second challenge problem here. Hari,
can you hear?
Yeah.
Okay, thank you everyone for joining. Now we would like to transition to the second part of this talk, which is basically on characterizing flow and pressure drop in complex geometries, spacer membrane geometries. Can you move to the next slide? So our aim really is to have a generic solver that can aid in providing models for the higher level systems models like ProteusLib. And so our as part of this HF effort, we have developed a solver using OpenForm, which is initially built on SUMS, which is Stanford's membrane solver. And with that, we have added features where we include variable properties, viscosity and density, and osmotic pressure as a function of concentration. And I would like to thank Professor Hoek and Professor Eric Hoek and Tim for providing us with some of these correlations. The one you see here on the plot on the left is from the OLI database that we use in our solver. Our solver is inherently incompressible, but it also accounts for the small density variations with the salt concentration, and it uses the Pimple algorithm for solving the Navier-Stokes equations. We also include bulk transport of solutes and variable properties. So mainly, the implementation of the boundary conditions is what is important here. We have a permeate velocity that at the membrane, which is a function of the membrane resistance and viscosity, and also it depends on the difference between the transmembrane pressure and the transmembrane osmotic pressure. The osmotic pressure is again a function of the concentration and also a salt rejection factor. Also, the next slide, I would like to describe what we do to model concentration polarization. Can you move to the next slide? So the CP boundary condition is essentially a zero flux for an ideal membrane, or a flux based on solute rejection on one end, as shown in the picture on the left. And what I show here on the picture on the left is a simplified one-dimensional approximation of what happens near the membrane, where you have bulk concentration away from the membrane, and which reaches a higher value at the wall. And this higher value at the wall is because of the flux boundary condition based on what is shown here in the set of equations here. And a film theory solution is easily possible by solving the 1D convection diffusion equation, which gives you a concentration polarization factor as shown here in the equation here. It again depends on the boundary layer thickness. Now, OpenFOAM implementation requires that there is a cell center value and a gradient data associated with the boundary condition at the boundary. So the initial way by which it was implemented, which is shown here in yellow, is as shown, which caused some negative values to appear at the wall based on this approximation. And this we kind of formulated this in a different way by using the concentration polarization factor equation, but using the dx by 2 as a length scale. And with this modification, we wanted to see if the accuracy of the solution is maintained, and that's what is shown here in a plot on the right-hand side, which shows the solution error, which is on the y-axis, as a function of number of cells. And I was able to show that with this modification, you would still get first-order accuracy using an upwind scheme for the concentration. And again, it's compared against the other formulation where the flux is imposed as a mixed boundary condition, as shown here. Okay, next slide. So we have verified our solver against several published results. The first one we did was to see if we were able to match pressure drops for a channel with very small thickness and porous walls. What is shown on the left-hand side is pressure drop against distance along a channel, which is about 2 meters long and 1 millimeter in thickness. And it compares well with published perturbation theory solution by Berman et al. In 1953 paper. The next thing we studied was the for a flat channel without spacers, 7 centimeters long channel, 1 millimeter thickness. And we looked at the CP factor variation along the length of the channel. And we have compared this with papers from Professor Eric Hoek's group, Subramani et al. Paper from 2006, and we were able to match the CP factor along the axis for several cases where we varied the permeate velocities through the membrane. Next slide. So as I mentioned earlier, really the aim of this facet of high fidelity modeling is really to get mass transfer correlations that can be applied at the systems level. So one of the first things we did was to see if our model was able to obtain the published correlations that I think is being used in ProteusLib, which is the Reynolds power 0.36 correlation for the Sherwood number. So I have replicated that case here from the Gillen and Hoek et al. Paper in 2009. It's basically a case with a rectangular membrane with cylindrical spacers. And the correlation is again found based on the equation for CP as shown here, where I find the CP from the simulations, which is the concentration polarization factor, and then compute the mass transfer coefficient from this equation and then apply it to the Sherwood number equation as shown here, where K is the mass transfer coefficient, DH is the hydraulic diameter, and capital D is the diffusion coefficient for the solids. And so with that, we were able to match what Guillen and Hoek had obtained in 2009 for the Sherwood correlation, as shown in the plot on the right-hand side. And one thing really that we observed was that this correlation is very sensitive to the grid resolutions. And this is mainly because the Peclet number for the solute equation is order of 30,000, while the Reynolds number is actually small. So therefore, to resolve the concentration boundary layer, you would need enough number of cells within the flow channel to obtain grid independent results. And that is what is shown here, where I'm showing mass transfer coefficient as a number of cells, as a function of number of cells along the channel thickness. And you'd see that you need about 200 cells really in along the depth direction, along the cross flow direction, actually in the depth direction, to obtain grid independent results. And this 200 cells corresponds to about a few microns, I think, in grid size. And that's what is needed really to obtain accurate results. And it again emphasizes the need for high fidelity modeling in these simulations. Okay, next slide. So with all these verifications done, now we moved on to doing some of these large scale cases. What I'm showing here is a case where we ran a 1 meter long axisymmetric membrane It has a diameter of 8 centimeters and a thickness of 0.5 millimeters. You can imagine this is like a 1 meter long newspaper folded into a toroidal cylindrical domain with a very small thickness, which is representative of what RO modules have as membrane spacer configurations. And what I'm showing here is a case where we have flow at about 0.8 meters per second. And I have also shown a zoomed in view of the vorticity field. And you can see that at this Reynolds number, the spacers are releasing vertical structures behind it. And the train of vertical structures is what is shown here. And if you look closely at the image, I'm not sure if it is showing up in the WebEx video. This is actually a movie where you can see tiny variations in concentration along the large scale cylindrical configuration. And these were done on NREL HPC. It was run using 1024 processors, and it required 2 days of wall clock time, and there were about 24.5 million cells in the simulation. And again, with this we are able to characterize things like what is shown on the picture on the right hand side for a cross flow velocity of 0.025 meters per second, where I'm showing concentration profiles along the axis as well as along the transverse direction towards the membrane. So the blue lines here show value of solute concentration at the membrane, and the red ones really show the concentration towards the middle of the channel. And you can see that there is clearly concentration polarization that is seen. The blue lines seem to have a higher concentration compared to the red. So it is able to capture CP along the membrane, as well as capture the increase in solute concentration because of permeation of clean water. Okay, next slide. So finally, we come to the main aim of this work, which is the mass transfer correlations at module scale. So the plot on the left shows the actual solute concentration profiles along the length of the channel. So different line plots here show solute concentration profiles at different positions along the axial direction. And in this case, you can see that it varies from 0 to 1 meter. And you can see that the solute concentration never gets to a fully developed profile until about 25% or 30% of the flow, and which kind of brings up to the important point where the correlations that I had shown earlier, the RE power Reynolds number or 0.36 correlations, were developed with an inherent assumption of a fully developed flow. And with that, I was able to generate the same Sherwood Reynolds number correlation plot for varying cross-flow velocities as well as for different points along the axial direction of a module. And due to the high Peclet number, again, as I had mentioned, the flow does not get to a fully developed profile until about 40% of the distance, or even it gets even longer for higher Reynolds numbers. And you can see that the profile matches in certain regions, but it does not. And these regions where you see these almost vertical lines of Sherwood number is basically in the region where the flow is not fully developed. That is, you see a range of Sherwood numbers for a given Reynolds number. So our future efforts now is really to develop these correlations so that Proteus Lib will be able to leverage it to obtain more accurate results. And I think the first step we will probably do is to get the Sherwood correlation as a function of a distance along the membrane. So instead of just Reynolds and Schmidt's power 0.36, we would have an X by L power factor, which could probably give a more accurate correlation. And that concludes this section of work. And I think we have just one more slide, which is basically the summary of all the work we have done as part of our first year effort in the modeling and simulation high fidelity task. We have identified important challenge problems, and with these, and we have made progress on each of them. Vimal has made significant progress on the high pressure side, and I have presented some of the work on the membrane spacer work. And now we will be able to open a dialogue with the other members of the NAWI community on designing better membranes and also aid in improving correlations in ProteusLib. And we hope that our effort is going to be impactful across several projects in the upcoming budget periods. Thank you.
Thank you, team. So I'd like to open the floor for questions from the audience. And I think as of now, you don't have an ability to unmute your mic, so you'll have to put those questions in either the chat or the Q&A. Are there any questions? So I guess I'll start with a question. Vimal, I'm curious, what was the most surprising result that you found in doing this modeling?
So the first, let me go back. So the first, well, a couple of things I would like to point out. The first is, so in these cases, we have most of if you look at some of the correlations typically used for porous media, like the Karman-Kozeny correlations, they do not consider the geometric parameters as one of the input factors. Like most of them only take the pore sizes, the equivalent pore sizes as an input to determine the effective permeability of these structures. But since specifically in these type of membranes, when these macrovoids are existent as a result of the manufacturing process, they are basically inherent in these membranes, and that significantly affects our estimates of the membrane permeabilities and membrane characteristics in overall in general. So so that was one of the important findings, or one of the important verifications we are able to do with this study. Basically, try to bring in more of the geometric parameters of these macrovoids or the membrane microstructures into calculating or estimating the membrane characteristics.
Thank you. So Ryan Kingsbury has a question. Will Proteus HF continue to focus primarily on CFD simulations, or is it envisioned that the platform will also encompass, example, sophisticated continuum transport models?
I can answer that. Even in the first year, we are actually planning to do a combination of flow and also compaction simulation. The compaction being, we could think of that as being having mechanics model in there. As the current scale of this is at the level of, when we say membrane, but we are referring to the mid layer, the support layer, where our thicknesses are on the order of microns, in this case, hundreds of microns, and about a millimeter thick. That's the length scale, where we are actually seeing this continuum flow model to still be valid and still be useful. But if we identify challenge problems that require modeling flow at different length scales, say going down, there we might require other approaches for modeling the flow. And that's actually a very good point. We might need to look at other ways for modeling flow through those systems.
Thank you, Ramanand. And Ryan Kingsbury has a second question. Have ProteusLib or Proteus HF been released on GitHub? And I can tell you that ProteusLib, which I assume was the first one, will be released in September in its first version. And how about Proteus HF, Ramanand?
In Proteus HF, NMAC foam, that which are that was used for the results that Hari presented. That's already available. That's being released on GitHub. The Quilt code has not been released yet. That's something that we are actually planning on doing, but has not happened yet.
Thank you so much. Okay, Stuart Bible wrote, who do you envision as the end user of this new software capability? Seems like it will require users to have extensive experience in OpenFOAM.
I can answer that question, at least from the OpenFOAM side. I don't know whether you would need extensive experience, but yes, you would need some experience in the use of OpenFOAM, at least as a user, less as a developer, I think. And another clarification is that right now, even NMAC-Foam is not publicly available, but we would be probably doing that towards the end of this project, which is the end of the year, October.
Right, and I think in our experience, generally, you're going to want to work with someone familiar with the high-fidelity modeling methods when you're using these tools. So Alondra Ramos has another question. On one of our earlier slides, you mentioned 3 focus areas for improvement, which will be prioritized going forward and why? Sorry, the inflection was wrong. Which of these will be prioritized going forward and why?
Was it the 3-4 challenge problems?
There are 2 challenge problems that we actually referred to. There are 2 other challenge problems that we identified, but we have not started working on those yet. Those are still planned work. And those challenge problems, the last 2, number 3 and 4, would be to inform better models in the process level model in Proteus Lib. But between 1 and 2, there is not a, we haven't come up with a prioritization or anything.
But as of now, these 2 challenge problems are the focus, and that is the prioritization of those 2 was chosen through discussions with the various elements of the NAWI program to find challenges that would be most helpful to resolve questions within the projects or the development of some of the tools. So thanks for those questions. We still have time for a few more questions if there are more questions. So I guess, Hari, I'd like to ask you the same question. What did you find the most surprising in the work that you were doing?
I think, yeah, the fact that the correlations were different, I think that was an important conclusion that we obtained from our simulations at the large scale. And initially, I and many of us had had a feeling that that may not be the case, and HF may not be useful at all. But now that we have done this, we are able to say that high fidelity simulations are important to identify those correlations. I think that's I think surprising and useful in some sense.
Thanks. So Tim Bartholomew, our collaborator, has a question. It's interesting to see how fully developed flow could be a bad assumption, which is what Proteus Lib does. To improve Proteus Lib, you guys suggested surrogate models rather than simple correlations. These surrogate models would be only applicable for a fixed membrane module size, correct?
Yeah, so again, I wasn't very clear on what I meant by surrogate models. It could be, a set of surrogate models that includes, one is basically the Sherwood correlation that, Tim, you may be using directly in Proteus Lib, or it could be that it is a surrogate model given a full-size membrane with so many spacers, and so it could include several parameters together to get to a surrogate model. But I think at least to get started, I think the Sherwood correlation is what I really meant as a surrogate model. But yeah, I hope that answers your question, Tim. I wish I could hear you.
Yeah, I guess point of order to the folks running this. Are we able to allow all the participants to use their mics?
Yeah, Deb, I can go ahead and unmute everyone, and then whoever doesn't want to be unmuted can go ahead and mute themselves again.
Okay, great. So Tim, if you have a follow-up to that, you're welcome to ask it.
Just give me 1 second while I go ahead and unmute the attendees real quick. Okay, everyone should be unmuted now.
Hi, this is Tim speaking. Can you guys hear me all right?
Yes, we can.
Okay, great. So, Hari, yeah, that makes sense to me what you're saying. Yeah, Sherwood correlations would be really easy to use on Proteus Lib. That's what we have it built for right now. As long as we have relationships for that. But if you go to your results slide, it was hard to see a relationship that we could fit to those things, right? I forget which slide number it was, right? But like, it was like each like case was different, right?
Yeah, you're right. Yes. Yeah, that is why the plot of Sherwood versus Reynolds is not accurate. And you would have to plot it in a 3 or 4 dimensional space to really see what is happening.
Okay, so you're saying weak function of Reynolds number, and that there's other things we need to. So this would, you would think other dimensions like the porosity or just the velocity and other things like that would make a better surrogate model?
That could be, but my real instinct here is that there is another factor, which is basically the distance along the membrane that goes along. Like there is an X by L, where X is the distance along the membrane and L is the length of the membrane. That would, give that factor to the correlation that relates to how well developed the flow is at that position. Is that possible in Proteus Lib? Do you have a?
Yeah. Yep, that's perfectly fine. Most Sherwood number correlations have that X by L, which you're probably familiar with. Yeah.
Okay.
Yeah, as long as we have a Sherwood number correlation from this work, then it can be applied into Proteus Lib. And we have like positional data in our 1D model. So if that changes by X, that's okay for us.
Okay, very good. Yeah, this will be great. And I would, I hope to talk to you soon about all this.
So this is Eric. Can you guys hear me?
Yes.
Hey, so it's really good to see how much progress you guys have made. Sorry, can you go back to the slide that you were just on? I'm not sure I understand how or why your Sherwood number is changing with Reynolds number the way that it is, these vertical sort of lines of different colors.
Yeah, so I can answer that question. So the Sherwood number is basically a measure of mass transfer coefficient. And mass transfer coefficient is basically the diffusion coefficient divided by the boundary layer thickness or the concentration boundary layer thickness. So if the flow is not fully developed, the concentration boundary layer thickness is changing along the axis, while the Reynolds number has stabilized because it is quite small. So for a given Reynolds number, I would see a range of Sherwood numbers because the boundary layer thickness has not stabilized. And that's why you see these vertical streaks I hope that makes sense to you.
How are you determining a boundary layer thickness?
It is from the film theory equation. So I compute the concentration polarization from the simulation, and then I use the film theory equation to get the mass transfer correlation.
So from your CP modulus, you're back calculating the mass transfer coefficient.
That is correct.
And then from there, D by delta is your mass transfer coefficient, basically.
Yes, exactly.
Okay, and can you go to your plot of, you had a plot of CP sort of along the length, I think.
Yeah.
So what you're showing here is the concentration increasing by a factor of, I don't know, 1.6 or 1.7, something like that, almost 2x.
Yes.
From inlet to outlet. But I think what's not clear to me, so when we calculate CP, we calculate the local concentration near the surface of the membrane divided by some value. What is the value that you're using to normalize that?
I'm using the value at the middle of the membrane to get CP.
So the center line,
Yeah.
the local concentration.
Yes. Isn't that what you use as well in yours?
Well, yes, but it doesn't look like that's what you're normalizing by. It looks like you're normalizing by the inlet concentration.
No, in this case, I'm actually plotting the actual solute concentration. So it is not normalized at all. So
what is this, the gradient in blue and red?
oh, that is so these are basically line plots at different different locations away from the membrane and close to the membrane. So 0 is really close to the membrane, so it is higher. The concentrations are higher, and towards the middle you see, lower concentrations.
Far away.
So you can imagine the thickness is what is characterizing CP.
Say that again?
The thickness of the set of lines that I show here, that really signifies how much CP you're seeing.
So is like the bottom of this plot sort of the red line that starts at around 32 and comes up to around close to 50? Is that like the bulk concentration?
That's the concentration at the center line, yes. If you average it out, it could be somewhere between the blue and the red. Yeah, so it's not the average, but it's the value at the center.
So do you show anywhere the value of the concentration polarization modulus?
I don't have it in this presentation, but I do have that, yes. And that again relates to the mass transfer coefficient.
Yeah, I'm just curious because you guys are modeling a pretty significant channel length. And so what we did in the past was for a small channel length, we took the length averaged sort of CP value. And then we used that to establish the Reynolds number. That automatically made it a simplified function, but your channel average, it's going to be hard to take an average over such a long channel.
Yeah. So we need to talk offline to see how we can make that.
I suspect that plot which had the Sherwood number versus Reynolds number with these sort of vertical lines, like that might be able to be collapsed into something which looks more reasonable depending on how you do the normalization.
Yeah, absolutely. I agree with that. Yeah.
That might be something to we can spend a little time on that if you want together.
Yeah, absolutely. I would love to talk to you again.
Okay.
Thank you.
Yeah, very cool. Thanks.
All right. Thanks everybody for the great questions, and thank you Hari and Vimal and Ramanath for the great presentation. And with that, we want to wrap up. But again, thank you all for attending this webinar. And as was mentioned, this will be available on the NAWI channel. And we don't yet have the August summer set, so I can't tell you what that is yet. But I do expect that in September, we'll have the team that's doing the analytics for the NAWI roadmap to actually present some of the work on the analytics in September. So if you're interested in presenting in August, please feel free to contact me, and we'd be happy to schedule you. Otherwise, we'll be setting that in the next week, and we'll let you know who will be that webinar. So thanks, everybody. Have a great day.